What is a pathogen? So, as we've already discussed, a pathogen is any kind of living organism that can cause or enter another living organism, you know, in most cases we'll think of the human, and cause damage to them, okay? So causes harm to that organism. The kinds of things that could act as pathogens are viruses, which are extremely small, this is not to scale, bacterial cells, which have cell wall, cell membrane, or prototists such as Plasmodium that causes malaria, or fungal cells, okay, which are, you know, eukaryotic and kind of are more similar to our cells, right? So again, this is not to scale. Now, there's some key things that we need to know about the terminology when we're talking about a pathogen. So the pathogen is the organism or the microorganism that could be coming into the host and causing harm. That's how we refer to either a virus, bacterial cell, or a fungal cell or protoctist. And the second thing is antigens. So we're going to be talking about antigens quite a lot. And antigens are any part of the pathogen that your body, your immune system could recognize. Okay, so this could be a particular protein, it could be a fragment of a glycoprotein on the outside of these molecules. It could be one of the proteins that make up the virus. It could be any bit or fragment of a pathogen that could be recognized by your immune system. Okay, so we're going to be talking about antigens, and I might abbreviate that to this, and obviously we're going to be talking about pathogens, but regardless of the example that I talk about, you must remember that I could easily just be talking about a virus, or I might as well be talking about a bacterial cell, or I might be talking about a protoctist or a fungal cell. A pathogen in general could be any one of these things, but I might use one of these as a particular example. In this, what I'll do is, in my little lecture, I'll be using the bacterial cell as an example, okay? So, your body's defense against these pathogens is in 3 stages, okay? Now, obviously the main stage is the immune system, but before that's activated, there are 2 stages that any pathogen has to cross before any of that becomes started, okay? So your first line of defense are your physical and chemical barriers. Okay, now these are like a wall that prevents any entry. So, you know, metaphorically speaking. So what we're talking about are things like your skin, okay, which is a very thick layer of dead cells, which is very difficult for any microorganism to cross. So that's a physical barrier. Other things are your gut. So remember, any food that you ingest, you're taking things from the environment and you're putting them into your body. So you have to be able to defend against any microorganisms that could be coming in, or pathogenic microorganisms that could be coming in at the same time. So what is your defense against that? So your saliva, first of all, has enzymes that normally digest food molecules, but they could just as easily work on carbohydrates found on microorganisms, for example, or proteins. Okay, so you have enzymes in your saliva. Your gut also has a very low pH. In that acidic environment, it could lead to the denaturing of the microorganism's protein, such that the microorganism is no longer pathogenic. It's not able to function without its proteins. Okay? You also have your respiratory tract. So when you're breathing in, again, what's to stop pathogenic microorganisms coming in the air that you're breathing? It's going into your body. What's your defense against any microorganisms in that air? So your respiratory system, or your respiratory tract, I should say, the lining of that contains two things. It contains goblet cells, which are producing mucus, okay, and that mucus traps any microorganisms that are coming in, but on its own that's not enough. Your respiratory tract also contains cilia. Now these are hair-like structures on the surface of the cells lining the tract, and they are constantly moving the mucus back up the respiratory tract, effectively removing the mucus that's got the trapped pathogens in it. Now that mucus can either be, coughed up and out, sneezed out, or, which happens more often, it's going to go down your throat and then to the gut, where it's going to encounter the low pH digestive enzymes that should destroy any pathogens that way. Okay? You also have your tears, which contain lysozyme, which is a hydrolytic enzyme able to break down the cell walls of the bacteria. There's also your sebum, which is the kind of oily substance secreted on your skin. That contains fatty acids, and they also provide a low pH environment for any microorganisms that could be on your skin. Okay? You also have the urinary tract in males and females. Now, the urinary tract, is also a point of entry for microorganisms. So, you have to provide the defenses in that area as well. Now, the urinary tract contain fluids which contain antimicrobial substances, microbial substances, right, as well as being acidic. Okay, so all these things are what microorganisms will encounter before they even enter into your bloodstream or your other important organs. Okay, so things like the gut, things like your respiratory tract. We think of them always as being they are inside our body, but they are still you still have to cross those things before you get into the bloodstream. All these things are kind of stopping the pathogen from getting into the bloodstream, into our important organs. Okay, so that's the first line of defense. Right, now, if something gets through that, and it is likely these are kind of non-specific ways to stop microorganisms getting in, but they're not 100% effective. Things can and do get through. So what happens next? So your secondary defenses are what the microbe or the pathogen will encounter if it gets through the primary defenses. And so basically that means it's got through, it's in the bloodstream, it's within the cells of our tissues and organs. What happens then? So if any pathogens do get through, they encounter the secondary defenses, and our secondary defenses are our phagocytes. Okay, now they are a group of cells which fall under the white blood cell family, but you don't call them white blood cells. Specifically, they are phagocytes, and if you want to name some examples, they are macrophages, and neutrophils. They do slightly different things, but they also have a property in common, that they are both phagocytes and they carry out the process of phagocytosis, going through the body, looking around on the off chance that it might encounter something that's not supposed to be there. Okay? Right, so macrophages and neutrophils, and what they do is if they encounter something that's not of the body, they will phagocytose that thing. Okay, so we'll think of that thing as being the pathogen, and let's just quickly talk about what happens during that process of phagocytosis. Okay, it's an important process. You do need to know the stages and key terms for that process. Okay, so we'll just continue in a bit. So, phagocytosis then, I'm just going to draw it in, right, maybe I'll just do it in one stage. So, phagocytes have a way of recognizing whether something is foreign or not. And if that thing is a foreign body, then it will be phagocytosed by the phagocyte. Okay, so that's our bacterial cell, for example. Okay, and this is our phagocyte. Now, what happens? So the phagocyte has got special compartments inside, or most cells do, called lysosomes, and they contain hydrolytic enzymes in them. Okay. Now these enzymes can, break down proteins, lipids, all kinds of things. So what happens is that when the phagocyte encounters the pathogen, what it will do is start to extend its membrane. And I'm just going to, show it in stages. It starts to extend its membrane so that the membrane starts to envelop, or, the preferred term is it starts to engulf the pathogen. Okay? Now, if this process continues all the way around the pathogen until the point where the two extensions meet and fuse with each other. Now, if that happens, you now have the pathogen within a vesicle of the phagocyte. Okay? Now, what happens next is these lysosomes move towards that engulfed particle. So I'll just draw the next stage now. So they move towards it. And by the way, just like these are called lysosomes, they're compartments in the cell, membrane-bound compartments in the cell. This new membrane-bound compartment in the cell is called a phagosome. Okay? Nothing too creative going on there, name-wise. So phagosome there, lysosomes there, okay? And what happens is that these lysosomes then move towards the phagosome, okay? And the membranes of the two compartments fuse together, similar to what happened there. When the membranes fuse, the contents of the compartments mix together. Okay, hopefully you can see that. Okay, and what that means is the hydrolytic enzymes that were contained in the lysosomes are now mixed with the pathogen, and this leads to the breakdown of the pathogen. That it's physically digested by those enzymes, and it's no longer harmful. Okay, so the pathogen now is broken down into lots of little bits. Okay, it's no longer harmful, its bits are there. And all these bits could act as antigens. Okay? They're all bits that your body could recognize and make a response to. So what phagocytes do, and what most cells that have pathogen in them do, is display some of these antigens on their surface. So this is the reason why the process of phagocytosis is very important, because it's essentially the step that starts your specific immune response. Remember, we're still talking here about the secondary defense. Okay? So, some of these antigens are then processed and sent to the surface of the cell, okay, and displayed outside. So, that bit displayed there, and that bit displayed there, and this bit displayed there. Now these are all different parts of the bacterial cell. Remember, it could just easily be a virus or a plasmodium, protoctist, fungal cell. But these are all parts, fragments of the bacterial cell which are digested and now harmless, but they can now act as antigens. Okay? And these antigens are displayed on the surface of the phagocyte in a process called Antigen Presentation. It's basically signaling to the body, look, we've got something in the body that's not supposed to be here. Here are its bits. We're looking for this stuff and trying to get rid of it. Okay? So all these different parts of the bacterial cell are displayed on the surface of the phagocyte in a process called antigen presentation. This can now also be called an antigen presenting cell. Okay? Right, so we've just finished looking at the secondary defense. Your antigen presenting cell is now displaying antigen on its surface. Remember, it could have been a phagocyte that has just digested a pathogen and is now displaying some of the fragments of that digestion on its surface. Okay, so bits of the bacteria are being shown around now on the surface. Okay. Okay, so the antigen presenting cell is presenting antigen, and what now comes into play is your lymphocytes. Now your lymphocytes are your specific immune response. So this is the third line of defense that's now being activated. It takes a lot of energy, takes a lot of resources to carry out this response, which is why you kind of struggle physically when you are mounting an immune response to an infection. It takes a lot of resources, takes a lot of energy during an immune response, which is why you feel so weak. when you have an infection, it's because your body's mounting an immune response. So why does it take so much energy? We're going to look now at that process. So first of all, you have a great number of T cells. I've shown 4, but it's more like millions. And each of those lymphocytes have a very specially shaped T cell receptor. Okay, now, there's a great number of these T cells because each of them has got a slightly different shaped receptor, and the system is trying to account for all the different types of fragments that could be entering your body, all these different types of antigens that could come in. Your body is trying to have something there ready to be able to complement that antigen and make a response to it. If you don't have the right shaped receptor, you could not make the response to that antigen, to that pathogen. So your body's got a great number, millions, of different T cells. You've got a few of each of those T cells, and each with a slightly different shaped T cell receptor. And what you want, so right now you have millions of different T cells, but what you want is millions of one of these T cells that could be the right one to fight against one of these antigens being shown. So here I've got 4 of them. Now of these, none of them would be able to complement any of the antigens, but we do have one that could. So this guy right here has got the right shaped T cell receptor to be able to bind the antigen, and that is what happens. So this guy, all of them will have a go at binding, but the antigen won't be complementary to the shape of the T cell receptors. It's a key point here. It's a theme that comes up a lot in this part of the course. So yeah, do mention it if you're answering questions on it. But the receptor being complementary to the antigen, kind of similar concept to an active site being complementary to the substrate. If the receptor is complementary, then that T cell can bind to the antigen. And when that happens, the specific immune response is activated. That's the system recognizing what it needs to make a response to, and it's a key step. This is called clonal selection, because we've selected one of the T cells out of the many millions as the one that could make a response to that antigen. Okay? So this stage is called clonal selection. We've selected, out of all the many millions of T cells, we've selected the one that could make an immune response, okay, in this way. So that's clonal selection. The next stage is expansion. So once you've selected the right T cell, that's great, but you've only got one. You need many of these. You need many of this one. So we need to clone this T cell, because we need lots of T cells with that receptor, okay, that recognizes that antigen that could be on those bacterial cells that could be multiplying as we speak. So clonal selection has happened. Now we are going to go through a separate stage called clonal expansion. Okay, clonal expansion. So once we've selected the right T cell, that T cell is now going to divide by mitosis many times, so that now we have a whole army of the right T cell. Before, we had lots of T cells, but they all had different shaped receptors, but now we've selected the one and copied it many times, okay? That's called clonal expansion. We now have a whole army of T cells that can seek out that antigen and kill any infected cell that's displaying that antigen. That is a very effective tool, okay? So, all these T cells, okay, we're not going to do all of it, but they're all T cells, and they have cloned themselves from that original activated T cell, okay? Clonal expansion. what also happens is differentiation, because you have all these T cells, but we do need T cells that are able to do slightly different things to make the most effective immune response. So as well as dividing by mitosis many times, there's also another process happening called differentiation. And that means, as well as expanding by number, you're modifying the T cells so that they are able to do slightly different jobs. And there are three types that you need to know about. First one is your cytotoxic T cell. Now this is the T cell that can, because it's got the right shape receptor, it can find any cell that is presenting that antigen, lock onto that cell and kill that cell. So its main job is to kill infected cells. How does it know that a cell is infected? Take a guess. The infected cells are presenting antigens. So these are antigen presenting cells. So as long as they're presenting antigen, that T cell, because it's got the right shape receptor, will be able to find that cell, lock onto it via its complementary receptor and kill that cell. Okay? Next, you have your T memory cell. Okay? Now, while the other T cells are short-lived, they are just alive, or they just exist in the time that it takes to carry out the immune response. Your T memory cells are long-lived cells, and they also have the right shaped T cell receptor, and because they're long-lived, they remain in the body for a long time, so that if you do become infected by the same pathogen again, which, if it does still have the same antigen, it will be able to quickly recognize it and more quickly undergo clonal expansion and differentiation again and mount a response again, but more quickly this time, because we haven't had to go through this stage again. We've already got that T memory cell there. Okay? And finally, you have your T helper cells, and the T helper cells have an important function in activating the B cell branch of the immune response. So far we've just been talking about the T cells, but there is a concerted dual effort between the T cells and the B cells, and we'll discuss a bit later why we need those two branches. Okay, so the T cells, which kill infected cells, your T memory cells, which retain the long-term memory of that infection and that antigen, and your T helper cells, which stimulate the B cell branch. Okay, the key thing here is that the T helper cells release kind of chemical messengers called cytokines, which help the B cell activation process. Okay. Okay, so we've just kind of reached this stage, through a number of different steps, we've arrived at a point where we've got an expansion of T cells, but as well as the expansion, the division by mitosis, we've got this process of differentiation, which gives us three types of cells, and we've said that cytotoxic T cells can kill infected cells. Okay, and we've discussed the role of the T memory cells, and the T helper cells is what is activating the other branch of the immune response, the B cells. Okay, so let's discuss now how the T cell branch helps the activation of the B cell branch of the immune system. So at the same time as T cells are being selected, you also have B cells which have to undergo their own kind of selection process and expansion process. So B cells can also display antigen, so they're displaying lots of different antigen, just like those antigen presenting cells were. But remember, they each would be displaying different types of antigen. So how do we select the one that we need to make a concerted effort between the two branches? Because you don't want the T cells acting on one antigen, whereas the B cells are working on something completely different. The effort needs to be focused on the same antigen to make it more effective. So you have the B cells which are displaying different types of antigen, and remember, your T cell by this stage is very specific for one particular type of antigen because it's been selected through the clonal selection procedure to have a receptor that has got a very specific shape for a very specific antigen. So these B cells are all displaying different antigen, and of these three, the T helper cell will recognize that B cell because that B cell is displaying the same antigen that was activating the T cell. So there will be complementary binding here, and at the same time the T helper cell will be releasing its cytokines, and it will help this particular B cell to undergo its own expansion. Okay, so these B cells will undergo their own clonal, so this process was their clonal selection. They will now undergo their own clonal expansion, and what will happen is that the B cell will differentiate into two types of cell. One, which is the B memory cell, again to increase the speed of response for a secondary infection by the same pathogen. And secondly, into a different type of cell called a plasma cell. Now the plasma cell has the job of producing lots of antibody, okay? And this antibody is specific to that antigen. So this is why the effort has to be concerted, because these antibodies now are acting on the same antigen that the T helper cells or the T cell branch is also focusing its efforts on. Okay, so you've got the T cell branch, which is mainly acting through your cytotoxic T cells to kill infected cells, and your B cell branch, once you activate the right B cell, and remember, it's not the B cells that make the antibodies. Once B cells differentiate into plasma cells then make the antibody that is complementary to that antigen. That's very important. Okay, so now you have antibody production by the plasma cells. So next, what we're going to look at is what antibodies do to remove pathogen from the body. So antibodies being produced by the plasma cells, remember? Okay, and because we've selected the right clone of B cells that differentiate into these plasma cells, they are all making antibody towards a particular antigen. But for that to make sense, we need to discuss a little bit about antibody structure. Now, I'm not going to go into the full details of this, I'm going to leave that to you, but I'm just going to talk about what's the essential part of antibody structure that you need to know for understanding the immune system. So there's well, let's begin from the basics. There's 4 polypeptide chains that make up the antibody. Now, if that makes no sense to you, it's because antibodies are proteins. We've discussed that before. Antibodies are proteins. They're made of polypeptide chains made of joined up amino acids by peptide bonds. 4 separate polypeptide chains that come together to form this functioning protein, i.e. it's got a quaternary structure. Those polypeptide chains are joined to each other by disulfide bonds, okay, which gives you this functioning protein. Now, this protein can be said to have 2 important parts to it. One bit is called the constant region. Now, this means that whatever antibody that you have, their constant regions are all the same, okay? So if I drew an antibody that was produced by a different plasma cell, it might look something like this, okay? So its constant part is the same as the other antibody. However, they have a second part which is called the variable region, and this means that for each different antibody, the variable region is different. What it does not mean is that bit can change. It can't change from that shape into that shape, for example. It's made with a very specific shape, complementary to a particular antigen. Once it's made, it stays that way. But for different antibodies, that part is different. Okay? i.e. variable. The constant regions are the same. Okay? So, that's the important part about antibodies right there. Now, what we have then is lots of plasma cells all producing that kind of antibody. Okay? And what we're now going to discuss is, well, what is it that these antibodies are actually doing? So what are antibodies doing then to try and clear the infection? Okay, clear the pathogen from the body. There's 2 things that you need to know about. Let's look at the first thing. So say if you have a pathogen, okay, it's got some antigens. Our particular antigen looked something like that, so that's also on the surface there somewhere. And now what could antibodies potentially be doing to get rid of this pathogen? Okay, so they're floating around in your bloodstream, soluble proteins, globular proteins, moving around in the plasma of your blood, on the chance that they will encounter the pathogen and be able to do something about it. And if they've got the right shaped variable region complementary to a particular antigen, they can do something. And we're going to look at what that is. So first, you've got our parasite there. Now, say if sorry, not parasite, but say if you've got a pathogen there, and say if it needs one of its proteins in order to be able to carry out the infection or to do harm to the organism in some way, what antibodies could do is, if they can bind a particular antigen that the pathogen uses to be infective, the antibody can actually bind to that protein or whatever it is and neutralize the pathogen from being able to use that protein to cause infection. So antibody binding to the right antigen, if the antigen is in some way important for the pathogen to be able to cause infection, by the antibody binding to it, it's actually preventing the pathogen from causing infection. That is called neutralization. Okay? That's one thing they can do. The other thing they can do is called agglutination, and it works on the same principle. If you have many pathogens, what these antibodies can cause, because they have two variable regions, they can bind multiple pathogens at the same time. They can bind multiple pathogens at the same time. That's Kyla. Kyla, you want to say hello to everyone?
Hello everyone.
Right, they can bind multiple pathogens at the same time and cause them to clump up. Now, a fancy word for that is called agglutination. Agglutination. And if you have a giant clump of these pathogens, it can actually help the process of phagocytosis to happen very much faster. Because the cool thing about antibodies is that, remember those constant regions that we talked about, the bit on the end that is the same for all antibodies? If any phagocyte is in the area, the phagocyte can actually recognize the constant regions through some receptors that it has of its own. So the receptors that the phagocyte has latch on to the constant regions, and this kind of triggers a response in the phagocyte that tells it that there's something here to phagocytose, and it begins that process. So the membrane starts to creep forwards and try and latch on to more antibodies. The membrane, will start to move on all sides, and it will move to try and bind more and more of these constant regions. And as you can see, if this process carries on, membrane creeps forward. If this process carries on, you can see how, if the two parts of the membrane join up, they will cause the engulfing and the internalization of those pathogens, i.e. the process that we've already looked at. Phagocytosis. So those circulating antibodies, don't forget that's where it all began, circulating antibodies latch onto the free antigen and cause them to clump up because they can bind more than one at the same time. And by making this large clump of pathogens that could all be phagocytosed at the same time by the phagocyte, it helps to clear the pathogen more quickly. Okay, so very quickly then, let's look now at how the T cell branch and the B cell branch are both working together to clear pathogen from your body. We'll go back to our example of the bacterial cells with those antigens on their surface that the immune system can recognize. Okay, so we have the free pathogens not in cells, but at the same time you have the cells of your body which have already maybe been infected, or the pathogen has already started causing damage in these cells. So say we have one infected cell here, 3 non-infected cells. You don't want to have any infected cells, and at the same time you don't want any bacteria to be able to infect cells. So what do? Firstly, we've already discussed that infected cells express the antigen, fragments of the pathogen that they've digested on their surface as a signal that something's wrong. So our cytotoxic T cells, because it has the right shaped receptor, can recognize those cells and initiate the process to kill that infected cell. So T cells, the T cell branch responsible to kill any infected cells, i.e. hopefully preventing further reproduction of the pathogen. Secondly, your free pathogens, not inside cells, what do you do about those? Well, your antibodies are taking care of them, aren't they? In the two ways that we've discussed, neutralization and agglutination. Okay? And we have our pathogen, which is using those antibodies to phagocytose those cells, right? So your B cell branch, via the plasma cells, creating antibodies directed towards a particular pathogen and clearing the free pathogen. Your T cells with the right T cell receptor, able to recognize the same antigen, clearing infected cells. Okay? And if this response is great enough and fast enough, you will clear the pathogen. Okay? Okay. Good luck, guys.